X-Talks connects professionals in the life science, medical device, and food industries with useful content like webinars, job openings, articles, and virtual meetings to help you succeed in your career. This podcast brings together some of our editorial staff to share insights into the latest B2B industry news to help keep you up to date.
Hello and welcome to the X-Talks Life Science Podcast. I'm Ayesha Rashid, Senior Life Science Journalist at xtalks.com, and this week I'm delighted to introduce a very special guest on the show. We have with us today John Celebi, who is the President and CEO of Sensei Biotherapeutics. Sensei Biotherapeutics is a clinical stage immuno-oncology company focused on the discovery and development of next-generation therapeutics for cancer. Through its tumor microenvironment activated biologics, or TMAP platform, Sensei develops conditionally active therapeutics designed to harness the unique acidic tumor microenvironment. John Celebi has over 25 years of experience building innovative entrepreneurial biotechnology companies. Previously, John served in varying leadership roles across various biotechnology organizations, including X4 Pharmaceuticals, Agenica Biotherapeutics, Arcule, and Teleric, where he conducted drug discovery and basic research for an antiviral drug program. John currently serves on the board of directors of Egle Therapeutics SAS, a biotechnology company developing first-in-class immunotherapies targeting immune suppressor regulatory T cells for oncology and autoimmune diseases. John received an MBA from Carnegie Mellon University and a Bachelor of Science in Biophysics from the University of California, San Diego. To learn more about John's work at Sensei in the arena of immuno-oncology, let's turn to him right now. John, welcome to the show.
Very nice to be with you here today, Ayesha.
Likewise. And so, John, I'd like to start off by asking you about your journey leading up to your role at Sensei. You've been in the industry for quite a number of years. And so what have you gleaned through your journey, and how have you been utilizing your experiences, your background in the work that you're doing and leading at Sensei now?
Thank you for that question. My journey was a journey in the sense that where I started is nowhere near where I am now. I started as a student in physics, as you noted in my background, and somehow I ended up in biotechnology, and it turned out that I loved it. I was at the bench for several years, and then I became a business person. I was doing a lot of business development work, then operations, and ultimately that led me to a CEO role. So what have I gleaned? I think you have to follow your interests. I think, I was never somebody who wanted to get their ticket punched at all the right places. I had a curiosity within me that needed to be satisfied. And at each and every role, if I wasn't following my interests and answering key questions, then that wasn't for me. I really trusted my gut on what I wanted to know and understand. And I think that led me to all the right places and brought me around a group of amazing mentors and people that supported my career along the way. So that would be what I've gleaned. How have I applied that to Sensei? I think because I followed my interests in each of these roles, it led me to a lot of different kinds of experiences. So, even though I only did 3 years of bench research, that connection to the science is invaluable to me. It informs my daily interactions with the scientific team that we have, and even the medical team that we have to some degree. In my business development roles, I was able to travel, to various parts of Asia and Europe, and built a lot of relationships abroad, which has led me to the board of directors of another European company. So those relationships have come in handy at various points in my career, and my operations experience as well, having really built a platform from scratch. So I think, if I were giving advice to a younger person who's just starting their career in biotech, I would say follow your interests, trust your gut, and things will work out.
Amazing. Thank you so much for that overview. And so, how did your interests lead you to the space of oncology and immuno-oncology? And at Sensei, of course, developing biotherapeutics that target components within the tumor microenvironment. So I'd love to learn more about Sensei's platform and its overall mission and goals, really, in this space.
Well, I think like many instances in my career, I've actually been lucky enough to been exposed to lots of different areas. In my case, I think probably a relevant question is, why did I stay in oncology where I've now been for almost 25 years? Because I started in antiviral research. I've certainly been exposed to other therapeutic areas, but my heart's always been in oncology. There's one thing that probably we can all relate to, which is we're all touched by cancer. And that's certainly true in my case. I've had family members and friends, but I think there's another level of scientific interest for me, which was really the complexity of the problem that we're trying to solve. The immune system is vast and sophisticated. And, with cancer, we're really trying to thread a needle because cancer cells are fundamentally different than our own normal cells in some way, yet they're derived from the same thing. And so what makes them distinct is very subtle. And that means we're really threading a needle. And I love that challenge. And I think that we've made some amazing progress in an industry that's full of really small incremental advances. And at Sensei, we've really tried to apply that focus and that understanding to our own platform. The tumor microenvironment is fascinating, and I think we're really just at the tip of the iceberg in terms of our understanding. But there are a few fundamental features we try to focus on at Sensei to take advantage of threading that needle that I mentioned. And one of them is the fact that these cells in the tumor microenvironment metabolize nutrients differently, which ultimately, through a series of various pathways, leads to a more acidic environment than normal tissue. And that's something we can draw a distinction on when we're targeting therapeutics to the tumor microenvironment versus normal tissues.
Yes, that's a very interesting approach because within the tumor microenvironment, the focus has largely been on the proteins or the oxygenation and so many other components, but to really look at the pH levels within that space, it's a very interesting approach. And so, what types of biotherapeutics is Sensei developing and to target or to as I understand, the biotherapeutics that are designed are actually designed to work within that acidic tumor microenvironment. Am I correct?
You're correct. Our expertise is in the antibody space. So that can mean anything from a traditional monoclonal to a bispecific, or even the use of those in other types of therapies, such as antibody drug conjugates, or even certain cell therapies, CAR Ts. Right now, we're focused in monoclonals and bispecifics. I think when we started this company, we really wanted to ask ourselves the question, how can we do things better? And not just different, not different for the sake of being different, but how can we take something tried and true, like an antibody, which has been used therapeutically for almost 50 years, and really have a different perspective on it. And so, when you think about, for example, the most successful immunotherapies, such as Keytruda or Opdivo, these are therapies that have had vast commercial success, impacted the lives of hundreds of thousands of patients, very likely, in oncology, and saved lives. But you look at the last 10 years and you ask, why haven't we had more successes like Keytruda and Opdivo? There really have only been 3 checkpoint drugs approved. Well, when we asked ourselves that question, we thought, well, these are all monoclonal antibody approaches, but they're the same type of approaches that people have been using for the last 50 years, i.e., antibodies are very specific. They home in on the target that they're designed to home in on. But in the case of the immune system, these are proteins that are found ubiquitously throughout the body. And so you now find yourself in a predicament where you're targeting a tumor, but you have an antibody that's really binding to normal tissues and the tumor. And sometimes you can thread a needle, like with the case of Keytruda and Opdivo, and have an acceptable safety profile and huge benefit. But in the case of many other proteins, that won't be possible. And that's actually what led us to our first target, which is VISTA, because that's an interesting case in its own right. So we found that by designing antibodies, that real needle in a haystack antibody that can distinguish the same target between normal tissue and tumor tissue by virtue of their differing pHs, that we could actually thread that needle in many cases and target the antibody even more selectively to the tumor.
And so in your pipeline, how many candidate products do you have? And also, what indications are you working in and evaluating your candidates in?
Our pipeline consists of 4 molecules. We have our lead program, which is now clinical stage, targeting the immune checkpoint VISTA. We have another program targeting a relatively novel immune checkpoint called VSIG4. A third targeting CD39, which has been challenging for the industry to drug effectively. And last year, we announced our first bispecific antibody targeting both VISTA and CD28. That program is unique because it's designed to activate co-stimulation of T cells by virtue of CD28. But that's been challenging because overstimulation of CD28 can lead to severe toxicity. And so our goal with this program is to drive a new generation of CD28 co-stimulation by virtue of its selectivity in the tumor microenvironment. And that selectivity comes from the VISTA arm of that bispecific.
And so what types of tumors are you targeting with these compounds?
Well, like most compounds in early development, we start very broad and then we narrow it down. And so we just completed, the dose escalation portion of our phase 1 study. And that's typically done in all comers, and that's what we chose to do. The next step was to narrow that down to a set of dose exploration studies. And for this, we did pick some specific indications, including lung cancer, melanoma, head and neck cancer, and also colon cancer, each for various reasons. And then the next step in our development process will be to pick 1 or 2 of those indications to really expand into either a phase 2 or a phase 3 type of study.
And so, having been in the space for 25 plus years, how have you seen, and what, of course, we've seen very many milestones, as you mentioned, the advent of immune checkpoint inhibitors like Keytruda. So how have you seen things evolve, and where do you see things evolving to in the coming years in oncology? I know it's a very broad kind of a question, but we talk about trends and with technologies and also the biology being better understood. So what are the types of things that are currently trending in oncology and immuno-oncology as well?
I think this is one of the most exciting times to be alive if you're interested in cancer research. It would be foolish of me to predict XYZ is going to work and ABC isn't. But what I would say is the pace of research being done right now, and I can say this based on my own research experience compared to 25 years ago, is light speed compared to what it used to be. And so you can process information more quickly, you can test things more quickly, you can produce material more quickly than you used to. And I think we've seen some really interesting examples of that in the last decade. I used to be involved in the field of antibody drug conjugates. Nobody wanted to touch them 10 years ago. And the last 3 or 4 years, with some really exciting clinical results. I think they've become extremely hot commodities again. And interestingly, immuno-oncology has been a field, despite the success 10 years ago with Keytruda and Opdivo that I mentioned earlier, has become really challenging. And so this is an opportunity now for people that are really committed to immuno-oncology to take approaches in new directions and try something different because the old approaches haven't been working. So I think we're going to see an era of continued innovation, of exciting clinical advances. I wouldn't be surprised if 200 or 300 years from now, people look back on the 21st century and say, this was really the era of biotechnology.
And also in terms of innovations and the evolution of the space, we also are seeing a lot more collaborations between different areas and stakeholders, for example, between academia, industry, and then patient advocacy groups. So is that something that you've seen also evolve over time? And does Sensei also participate in those kinds of collaborations?
Well, what has stood out to me is that it is so much easier to run a company with a smaller group of people than it ever used to be. When I started in the industry, you had to build everything yourself, and so you just didn't see as many companies getting off the ground. But there are so many high quality service providers, CROs, CDMOs, et cetera, and a network of relationships that you can really get a lot done with smaller groups of people. I think the model between academia and industry has also evolved. I'm not as familiar with that because we've really embedded all of our IP ourselves, but I have seen some really productive collaborations between industry and academia, and mostly that's because the academic side of things has really evolved. And I think if you talk to an academician, you'll probably hear quite a debate over how much they should be involved in industry. But from the standpoint of developing therapeutics, there's no question it's been beneficial overall.
And of course, there are a number of unmet needs in oncology. In your opinion, what are some of the most pressing ones? And how is Sensei kind of positioning itself to address some of those pressing unmet needs?
Well, cancer treatment has changed dramatically over the last decade. So you have now patients, for example, across many indications, including lung cancer and renal cancer, who are often treated with immunotherapy as a frontline therapy. And some of those responses are extremely durable, meaning they can last for 6 months or more. The question is, once these patients acquire resistance to these inhibitors, what comes next? And so to me, that is a huge area of unmet need. And it's a practical question too, because it's no longer possible to run a clinical study for experimental therapies with checkpoint immunotherapy naive patients because they simply don't exist in most cases. And so really understanding what drives resistance to checkpoint inhibition, whether it's acquired or it's innate resistance, is a fundamental question. That's the question we're focused on at Sensei, and it's actually what led us to Vista as our first important target for clinical development.
And I want to just go back to Vista, actually. What makes this such an important and a key target? It's actually not a target that we hear too much about, but I'm very much interested to learn a bit more about how it plays a role in immune function and in T-cell regulation.
I think there are a few aspects of Vista that make it absolutely fascinating. And you might be surprised to hear that it actually comes from the same family of proteins as PD-1 and PD-L1, which are the targets for Keytruda and Opdivo. That's the B7 family. And so that tells you that it has some structural similarities to those proteins and probably plays a role in suppressing the activity of T cells. And indeed, work over the last 10 or 15 years has shown that it is highly immunosuppressive. The interesting thing about VISTA, well, I shouldn't say the interesting thing, there's a couple of interesting things about VISTA, is it is distinguished by the fact that it is primarily expressed not on the tumor, but on these other immune cells that are found in the tumor microenvironment called myeloid cells. And myeloid cells have been known for quite some time to play some role in immunosuppression. In fact, there's even this term that is used that I'll be criticized for bringing up, which is called myeloid-derived suppressor cells. There's probably a lot of academicians out there that would not like me using that term. I use it here mostly to point out that myeloid cells clearly have immunosuppressive function, but the mechanism isn't always that clear. We believe that VISTA is one of the mechanisms by which myeloid cells exert suppression on T cells. And there's clearly increased expression on these cells following a checkpoint expression. The second thing about VISTA, which I find totally fascinating and I think is something that needs a lot more work, is that VISTA itself only becomes a checkpoint, only has checkpoint function under more acidic conditions. That makes it a perfect first target for our platform and our purposes. But you have to ask yourself, why is that? And if VISTA has this very sensitive pH type function, are there other molecules out there? There almost certainly have to be, that are playing a role in a suppressive tumor microenvironment. Also, cancers typically don't invent new biology. They sort of take existing biology and dysregulate it and adapt it for their own purposes to proliferate. So what is the role of VISTA in normal tissues under low pH conditions? And is there a network, kind of this low pH network of crosstalk between proteins? It's totally fascinating. And I think this is a subject of our own research. It's beginning to burgeon up in the academic field. And I bet there are a lot of other targets out there that operate in this way.
That's very interesting because when I was first learning about Sensei ahead of this interview, and in your approach in targeting, or rather developing therapeutics that function optimally within a specific tumor microenvironment that is acidic, I thought about the bone marrow microenvironment, and so when you mentioned that VISTA is typically more highly expressed on myeloid cells, so there's that connection there. So is the suggestion that perhaps regardless of the tumor type, that maybe focusing in on that bone marrow microenvironment is important because that is essentially where all of your immune cells originate from, and so taking a step back and going into that microenvironment, or myeloid cells can be fine. They differentiate obviously into monocytes and different cells later on in their lineage, but is the bone marrow microenvironment somehow unique in terms of being a therapeutic target, not just in leukemias, let's say?
It's a question that's near and dear to my heart, as my mom is fighting multiple myeloma as we speak. I'm
sorry to hear that. Yeah.
So this is an important question, and one which woefully we do not know enough about. Mm-hmm. But in talking to experts, you're 100% correct. It is known that bone marrow has a generally speaking, slightly lower pH than normal tissues. And so one could hypothesize that under the conditions of abnormal proliferation, such as in multiple myeloma, acute myeloid leukemia, other diseases, would it drop even further? This is a question we're asking ourselves right now, because in that case, antibodies like ours, SNS-101, which is targeting VISTA under low pH conditions, could also potentially be applicable.
And so what can we expect in the coming months and coming years at Sensei in terms of your developmental pipelines and key milestones that you're targeting?
Well, this is a very big and exciting year for us. We've been about a year into our phase 1 study. We're actually announcing our first efficacy clinical results later in Q2. So that's a big event for us. We also recently launched an expansion to our study, which is going to be testing, really confirming doses and also looking at safety, PK, and efficacy in other patients. And we also plan to have an end of phase 1 meeting with the FDA later this year. So this year, a large portion of management's attention will be directed towards preparing for that FDA meeting and also defining the paths that we intend to take to maximize the value of our lead program and our other preclinical programs.
Excellent. Well, we wish you all the best in those upcoming milestones, and really looking forward to seeing you reach those milestones in the near future. And we'll definitely be keeping our eye out and keeping up to date here at X Talks. So, John, I'd like to thank you so much for your time today. I really appreciate it.
Thank you, Ayesha. It was a pleasure to be with you today.
Likewise. That's the end of this episode of the X Talks Life Science Podcast. If you liked today's show, don't forget to rate, review, and subscribe. Thank you, everyone, and we'll see you next week. Bye for now.
If you enjoyed our discussions today, please share the episode with your friends and colleagues, and be sure to subscribe in order to be notified when a new episode is released. To join in on the discussion, you can find X Talks on social media, email podcast@xtalks.com, or comment on the articles directly. Links are in the show description. Take a moment to join our community at xtalks.com to get access to everything we have to offer, including webinars, job listings, virtual meetings, articles, and more. The views and opinions expressed in the podcast are those of the speakers sharing them. They should not be taken as professional advice and do not necessarily reflect the policy or position Honeycomb Worldwide. For further information, email us at podcast@xtalks.com. Thanks for joining us, and we'll see you next week.